speakers, and I'm obliged to all of you for accepting my invitation and sparing your valuable time for my students. And my dear students, you will glad to know that Dr. Ajit Bharti and Dr. Sharmista are both from Jharkhand, and from not only from Jharkhand, but from our university, that is Ranchi University only. So I hope you'll be able to make the best out of this opportunity. I'm glad to find that a number of students have joined this program. So I welcome you all here.
Thank you, Madam Principal, for enlightening us on all the topics of web lecture series, Recent Trends in Preventive Biology. I'm Dr. Suma Roy from Department of Biotechnology. Good morning, sir. Welcome once again. And good evening to dignitaries, colleagues, and participants on the first day of this 3-day International Web Lecture Series. We are honored to have with us Dr. Ajit Bharti, Assistant Professor, Department of Medicine, Boston University Medical School, USA. The most notable part of his introduction is that he is an alumni of Ranchi University. Dr. Ajit Bharti did his PhD with Professor Mehrotra from Ranchi University. During his dissertation, he worked with Dr. Feroz Ahmad in Ranchi University and Dr. K. Murugesan at AIIMS, New Delhi. Postdoc at AIIMS with Dr. Murugesan for 1 year. He joined DW Caves Dana-Farber and 4 years, continued work at Dana-Farber Cancer Institute and Harvard Medical School for another 4 years. He joined Boston University Medical School in 2002 and is at present working as assistant professor in Department of Medicine. Today, he will be giving us an amazing vision on precision medicine, the core principle of which is to decoding the genetic and molecular changes that of disease. We can all focus on his laboratory is to understand the cancer drug-resistant mechanisms, particularly topoisomerase 1 inhibitors. He has started 2 biotech companies in the areas of drug discovery. Dr. Bharti's laboratory defined the mechanism of resistance, identified topoisomerase 1 PS10 as the key molecular determinant of DNA damage response as a potential predictive biomarker for his class of drugs. The energy of topoisomerase 1 PS10 as a predictive biomarker will significantly enhance the clinical utility of topoisomerase 1 inhibitors and will change the response rate in the population. He has published 50 research papers, some in high impact factor journal, including Nature. Welcome, Dr. Ajit Bharti.
Can you hear me now? Can you hear me?
Sir.
Yes.
Yes, sir. Yes.
Okay. So I would like to share the screen first so that we can just start. Where do we share the screen? You might have to help me out here. Okay.
The screen is visible, sir.
Can you see my screen?
Yes, sir. It is visible.
So can you see my first slide?
Okay,
Yeah,
sir.
it's visible. Yeah.
So let me start with thanking Dr. Nihar for the invitation. I feel very privileged and honored to be the part of this seminar series. I would also like to thank all the faculty and the students. I would like to interact with them. That's a distinct privilege for me. And also some of my friends who kindly joined me at the last minute invitation. And I'm also looking forward for making it a little more interactive. I will request to put your questions whenever you want. It's not a very kind of a scripted kind of a presentation. It's very kind of a presentation where I can be interrupted. So let me declutter some of my jargons in this topic. So when we say precision medicine, thing is, can we predict before treating the patient whether the patient will respond or not? Right now, we treat in the cancer area, and I will be discussing mostly cancer. Only 20% of the patient get precision medicine. They know only 20% of the patient know that they will respond to therapy or not. So, so this is still one of the biggest challenge in the clinical oncology. So we were working on basic biology. We started working on topoisomerase 1 inhibitor almost 25 years ago, and when we started in 2002, we asked one simple question: how topoisomerase 1 is regulating the cells? And we ended up developing a test that will predict whether the patient will respond to this class of therapy or not. So this, sometimes we call it translational research, you know. And it's an old drug. Topo inhibitors were approved by FDA in 2002. So it was an old drug, but recently a new class of drug has been developed called antibody drug conjugate. In that one, the payload of topo 1 inhibitor is working very, very well. So we find ourselves in a very new position with a new meaning with our work. So let's start with our simple. I will be telling some of the stories in between to make it a little bit less compact presentation. So if you remember, then you all remember that DNA is a double helix. And I will also tell you some of the stories of the major breakthroughs in the technology that helped us to develop our story in the last 25 years or so. So when DNA double helix was proposed by Watson and Crick, it was not an easy sale. No one accepted it initially. They were young, in their late 20s or something like that. And Linus Pauling has already proposed, Linus Pauling, who was the Nobel laureate at that time, he has already proposed that DNA is a triple strand structure. And you can imagine the intensity of research people were doing that time to understand the DNA structure. And one of the things that was discussed is, okay, if it is a double strand or triple strand, how it is going to replicate. If you imagine a 5 feet long stretch of DNA is compacted in the nucleus, how it replicates. It's not an open-ended double strand structure. Whenever you do the replication, it will create supercoiling at both ends of the DNA. The supercoiling can be so immense that it will not let the transcription or translation or replication process continue. So one of the question was how the supercoiling is resolved in the cells. At that time, there was only one person who can cut and ligate DNA. His name was James Wang, the right-hand figure photograph. left-handed James Watson. I had an opportunity to listen to Watson a couple of times during my stay in Boston. He recruited, so Watson came from England and then started working in Wisconsin, but soon he was given a faculty position in Harvard and a lot of resources to develop one of the best molecular biology facility of the world. And so they started looking into the possibility how we can prove that DNA is a double strand. So how do the DNA double strand resolves its supercoiling or its kind of knots and rings and things like that, because it's very, very compact in the nucleus. So he recruited James Wang. James Wang was one of the very few people who can cut and ligate DNA, and he's the one who purified a protein from bacteria which can cut and ligate DNA, and he named that DNA topoisomerases. I had a opportunity to meet him. We got some DNA and other reagents from his lab, and I'm still in contact with him. So I will be discussing. There are a variety of topoisomerases in different species, but I will be discussing mostly topoisomerase 1. And one of the reasons for working on topoisomerase 1 is when I joined Dana-Farber Cancer Institute, there was a clinical trial going on of a cancer drug that specifically target topoisomerase 1. So we started doing basic biology as well as clinical trial. So if you see the linear structure of topo 1, it is a 765 amino acid. I would like you to really look into the different domain of this. N-terminus domain is poorly conserved, it is highly charged, and it is unstructured. The crystal structure of this region has not been defined yet or developed yet. Unfortunately, this is the part where I am interested to work on, and we basically got some of the data that we are looking into this area more carefully. There's a DNA binding domain that is highly conserved. Even amino terminus part of it is very conserved in the different species. The C-terminal is the enzyme or catalytic domain where there is a tyrosine 723. That is critical for the enzyme activity. So as I mentioned that topoisomerases cut and ligate DNA, this tyrosine 723 is critical for cutting and ligating DNA. If you look into the lower side, this is the crystal structure of topo 1 binding DNA. It basically circles around, wraps around the DNA, and 723 can cut and ligate. And the mechanism that works to reduce the supercoiling goes like this. If you see the upper part here, if you see the transcription and replication fork usually progresses in this fashion. And since the DNA is bound to some surface or nucleosomes and things like that, it will create supercoiling like this. What happens is when 723 tyrosine of DNA can form a covalent bond, it will cut and form a covalent bond with the DNA backbone. And then it will religate by the same transterrification reaction. Between cutting and ligation, there is a controlled rotation that reduces the supercoiling, if you can see in the middle panel. So these models are pretty well established. What happens our interest was not in looking into the supercoiling reduction and all those things. We were looking into the drug mechanisms. And topo 1 inhibitors were developed in early 70s by plant extract screening. NCI was taking a special program to make extract from the plants and try to find out novel anti-cancer drug. In that process, they found two drugs from the plant. One was camptothecin and the other was Taxol. Mansukhwani and Monroe were the two people who discovered both the drugs. Camptothecin turned out to be very toxic. So after phase 1 trial, the whole program was terminated in late 70s. However, in 1985, a group found out, and NCI used to distribute the compound to find out the mechanism of action of these compounds because this was a very potent drug. So a group in working in Johns Hopkins, they found out that the specific camptothecin is specifically targeted to topoisomerase 1. Then a couple of drug companies started making analogs, and now first generation topo1 inhibitors were camptothecin. Second generation, they made different analogs of topotecan and irinotecan. And third generation drug was nano-linked drug that just got approved like 5 years ago. And so topotecan and irinotecan are used extensively for variety of GI cancers, gastric cancer, colorectal cancer, pancreatic cancer. There are some third generation other drug that is non-camptothecin inhibitor, and that is yet to come in the clinic. But first three, second and third generation drugs are in clinic and used extensively to treat solid tumors mostly. So what topo1 inhibitors does, how it works, topo1 inhibitors are not enzyme inhibitors. They are DNA protein interface inhibitors. What that means is when the topo1 cuts the DNA, it forms a certain steric structure where it is prone for having and if there is camptothecin around there or analog like irinotecan is around there, it gets stuck to this structure in a way that religation is stopped. So what I'm trying to say here is topo1 can cut the DNA, but in the presence of camptothecin, it cannot religate because of the steric hindrance, not the enzyme inhibition. And interestingly, the covalent bond of topo1, tyrosine 723, get stuck. So you can imagine a situation where the topo1 is wrapping around DNA, cutting one strand of DNA, and covalently linked. When the replication fork collides with this nicked DNA, it causes DNA strand break. Now, just envision for a second that there are a million of topo1 which are cutting and ligate replication fork. What will happen is DNA topoisomerase 1 cuts only in one strand, but when there are millions of molecules, they can cut either strand. And a collision of replication will create a DNA double strand break. Now, unless this is repaired, DNA double strand break will cause cell death. So theoretically, it should work in all the dividing cell that is, and we know that cancer cells divide all the time. But only 13 to 30% patients respond to this class of drug. So early on, there was proposal that MDR or transporter-dependent resistance mechanism are involved. That means, what this means is there are ABC transporter proteins which are on the cell surface. They can pump out the molecule. Later on, it turned out that CPTs or topo1 inhibitors are not the substrate for ABC transporter. Topo1 mutations were proposed that if the topo1 is mutated, the drug will not. But their topo1 mutations were found in cell line, but not in patient population. So this theory was also discarded. DNA damage response pathway, they try to find out what are the genes specifically that are involved in DNA damage response pathway. They are impaired. But DNA damage response pathway, they didn't find any specific gene by doing siRNA library screen and all those things. None of the gene, a particular gene was found to be responsible for providing DNA topo1 drug resistance, inhibitor resistance. But early on, actually, from 1995, there were some publication that was telling us that it is the rate of topo1 degradation that will determine the response or non-response in the patient population. So this is the cartoon representing topo. I was working with Eric Rubin, and we saw that we were getting the patient's blood from the clinical trial sample, the leukocytes, and looked into the protein topo1, and we realized that topo1 is, even in the leukocytes, the topo1 level is down. The same year, there is another group at Yale University, they showed that topo1 level is reducing even in cell lines. They had the antibody, actually, we got the antibody from them. Then Desai et al. In a series of publications showed that topo1 is degraded by ubiquitin proteasomal pathway. And the rate of degradation determines whether the patient will respond or not. So when I started working in 2002, that time, this much was known that the rate of topo1 degradation might be the key of camptothecin response. So we didn't looked into the ubiquitin proteasomal pathway, nothing. What we did is we asked what are the proteins that are binding with topoisomerase or topoisomerase 1. So if you look the panel A, it's a very busy slide, but I will take a minute to really explain this because this is the key slide that we started in 2002, and this was the basis for the last 20 years of my work. So if you see this, these two lanes are indicating the proteins that are interacting with topo1. We had developed a specific assay that we grew like liters of cells and tried to find out what are the proteins binding with topo1. This kind of experiment I did with Eric also, but that protocol was not good enough to isolate the whole protein complex. And we used to we were getting one protein, and we did publish one topo1 interacting protein called nucleolin. But here when we saw this very clean, these are our negative control lanes, last two lanes. So this one was pretty controlled. Control was not having any binding protein. What we are seeing dark here lines are proteins that has been stained with silver staining or Kumasi stain. You can resolve the protein on a SDS polyacrylamide gel and stain the proteins. So this one was really a very amazing. We tried to reproduce this. This gel took us 6 months, a good 6 months time to reproduce and re-reproduce. And we were very happy that we are getting a very specific set of proteins that are binding to topo1. Now, the other challenge was how to identify these proteins. You know that they are your proteins, but how to identify them? In 90s, when we were working to identify protein, it has to be chemically digested from the amino terminus. So if, suppose we take this band, protein band, and go to the chemist, or we had a microsequencing facility, there we will go and ask them to identify this protein. And they will take a week time, maybe even longer, digest it from the amino terminus side. And 90% of the time, amino terminus was used to be blocked. So I remember very clearly when we got our nucleolin identified, our core facility, which can do the protein identification, was in the same building where our lab was. The difference was the microsequencing facility was on 8th floor and our lab was on 16th floor. When Eric found out that topo1 is binding with the protein nucleolin, when he went to talk with the senior technician, he basically ran all the way from 8th floor to 16th floor on stairs, basically. He was so excited. Why I'm talking about this is now the life has been changed. And I want to know the younger generation that how the new technologies have basically put the research, biomedical research, in a express lane, really. Imagine these 8 proteins are not as abundant as my proteins were in 90s. But by the time when I got this gel, there was an instrument called mass spectrometry that came into the biology lab. That time I was going to Dana-Farber to use the mass spectrum. I was trained in using mass spectrometry. mass spectrometry is a very old technique, but for small molecules. But for large molecules like protein, it is very, very challenging. It was very challenging till around 1998, when someone developed a protocol to digest this protein by trypsin and throw in the mass spec into a small peptides and ask what these peptides are. Well, we can find out what the peptide mass is, because mass spectrometry will give mass, not only mass, but mass by charge. So mass will be, for example, 180.345. There will be another peptide for the same protein, which will be having a different molecular weight. So you generate a peptide mass fingerprint, and then you go to database and ask what this protein is. Till 2002, till 1999, when the human genome was not sequenced, we go and find nothing basically, because in the database, only 2,000 proteins were known. So our mass fingerprinting search used to come with 4 or 5 proteins, most of them from HIV. Because HIV whole genome sequence was put in there and all the proteins were identified. But as soon as, literally, as soon as the whole genome sequence was put into public domain, there were people waiting in the mass spectrometry big facilities. They worked overnight to translate that into proteins and put in the database the mass fingerprinting. And believe it or not, all these proteins I was able to identify in one day. So these are the things that mass spectrometry changed the way we were identifying the protein overnight. So we identified these proteins as Q70, Q80, BRCA1. And I'll not go into the detail what these proteins do and what not, but they play a very critical role in DNA damage response pathway. So it was making a lot of sense to us that this is the way to move forward. By that time, it was also known that Q70 and Q80 binds and forms a dimer with another kinase called DNA-PKCS, DNA-dependent protein kinase. That made even more sense, because this heterotrimer is a very critical molecule in sensing the DNA damage. So they are not only the sensor, but they are also a stabilizer of the broken strand. They hold like a flower basket, literally. The DNA strand can zip through their top of the ring-like a structure, and they will stabilize this broken strand, and they have the mechanism to ligate them by force, literally. They have 4 or 5 other binding proteins. So when we knew that Q70, Q80 is binding, we first asked if DNA-PK is also in the complex, and we found out yes. So Q70, Q80, and DNA-PK was binding. DNA-PK is a very active enzyme when we give them a nicked or broken strand of DNA. So one of the first thought came that, okay, if they are binding, are they phosphorylating topo1? DNA-PK is a serine-threonine kinase. That means they phosphorylate proteins at serine or threonine. So again, the mass spectrometry came for my rescue, and we found out that serine 10 is phosphorylated by DNA-PK. When we found out this is the mass spectrum, one of the mass spectrum that we generated with one of our collaborators at MIT, Forrest White, because we tried ourselves, it didn't work. For that time, the early part of our mass spectrometry does not behave very well with the phosphorylated peptides, because phosphorylation causes negative charge on the peptides, and negative charge peptide do not fly very well into the mass spectrometry. So if you see the G, panel G, serine 10 is conserved right from Homo sapien to bacteria. And we were like, this data was like, oh, this is real, and we are on to something right now. So from this slide, I will take that we have isolated a protein complex, and we identified some of the protein that plays role in DNA damage response pathway, and topo1 inhibitors cause DNA damage. So it was making all the sense. And serine 10 is phosphorylated, and serine 10 is very conserved. And one of the more exciting part in this gel A was that topo-1 binds with BRCA1, BRCA1 also. So we will talk a little bit more about that in a later, in a minute. So serine 10 is phosphorylated here. And if you see this, what is the function of Q70, Q80, it was known by that time that they formed the non-homologous end joining. They repair the DNA by non-homologous end joining. And this is the model that was proposed that time. Now it has been very modified, but take-home message is we were in a right track to see if this association between topo1 and Q70, Q80 can make sense. Some of the people say that Q70, Q80 are very sticky protein, it binds to everything, but we still pursued basically, you know, but we hit the roadblock very quickly. The problem is this: we got and it plays very critical role. It provides attention. So DNA PKCS, we knew a lab at Children's Hospital in Boston. They had all kind of reagent on DNA PKCS. Fred Alt is the person. We requested him, and he was kind enough to give us a cell line where there was no DNA PK. It's a skid cell line. These mice are very prone for infection. It's a mice skid cell line. And they had, what they had done in the same background, they had added back DNA PK. So if you see the left-hand panel, in the top left-hand panel, CSV is a DNA PK deficient cell line, and right-hand panel, SCH8 is DNA PK efficient cell line. So we took this cell line and found this particular signal is DNA. In CSV3, the full-length DNA PK is almost not there. We have a PK. Into this, then the question is in protein level, in the middle panel here, there is no difference in the protein level at all. We looked into the activity of topoisomerases. This is the classic assay of topoisomerase activity, where you relax the DNA, and once you relax the DNA, their mobility in the gel is reduced. So, and isomers, these like the ladder-like structures are the isomers based on the relaxation of the DNA. The more relaxed DNA, they will run much slower compared to very supercoiled DNA here. They will run faster. So this is the assay. We didn't see any activity change in topo1 structure function. Topo1 amount, topo1 activity, and we were struggling to figure out what to do. But a couple of young students who came from, they were undergraduate students. I was just setting up my lab that time. And if you see the lower panel here, left-hand side, we treated these cells with topo1 inhibitor. And you can see right away SCH8, which is DNA PK positive, they degraded topo1 rapidly. In 6 hours, almost all topo1 was gone. Topo1 protein was gone. In SCSV, it was not degraded. Not much, at least. If you see, even in 6 hours, you can see the signal. The lower panels, whatever you see is, we always run a beta-actin as a protein, as a control for the load. This indicates that we have loaded in this gel similar amount of protein. So this was like a aha moment for us that, okay, now we know that without DNA PK, the topo1 will not be degraded by topo1 inhibitors. So this was the one line of evidence, right? We wanted another line of evidence. The other line of evidence was DNA PK inhibitor was developed by a company with the idea that they can develop it as a drug. If you see the last panel here, this is the control where it's a control cells. Most probably these are HeLa cells. And in the middle panel, if you treat the cells with topo1 inhibitor in the panel 2, here, you degrade topo1 completely. Since of PK, it is degraded. So these are two lines of evidence that gave us really good confidence that the topo1 degradation in response to topo1 inhibitors require DNA PK dependent phosphorylation. So now, from in the first, we saw the model directly going here. We changed this cartoon to this now. And now we are saying that topo1 will be degraded, but it will require DNA PK to phosphorylate topo1 at serine 10. And this really opened up a lot of avenues for us. One of the things that we early on did is started making antibodies against this serine, phosphorylated serine. And I will show you some data why we did that. I had a collaborator on DNA PK area for some time, Susan Listmiller in Canada. Not right that time, but after 4 or 5 years when I had already patented all these things, I talked to her one time and she said that they had done a different, they have identified this phosphorylation by a very different protocol or very different technique where they concluded that this DNA PK, her lab is a DNA PK lab, one of the top-notch lab in the world. She says that serine 10 is the highest phosphorylating peptide they found out in their assay. So that was a big validation for me, and we started thinking big on this one. So now we have modified from 2-step topo1 degradation to 3-step. So what I'm trying to say is we started finding out the molecular determinants of topo1 degradation with the potential of this phosphorylation of topo1 serine 10 can be used as a predictive marker. So how does the ubiquitin works? So ubiquitin proteosome pathway requires 3 sets of enzymes, E1, E2, and E3s. E1, there are only 2 E1 in the human genome, ubiquitin activating enzyme E1. There are now around 3 dozens of E2, but there are more than 100 E3s. And E3, so if the protein has to be ubiquitinated, we need 3 sets of enzyme that will transfer a peptide of 78 amino acid peptide called ubiquitin and conjugate that one to a protein that is supposed to be degraded. E1, E2 they do not have selection mechanism. They do not determine a specificity. But E3, since there are hundreds of E3, they determine a specific substrate of ubiquitination. And by that time, it was known that BRCA1 is an E3 ligase. So we have 3 sets of enzyme, ubiquitin activating enzyme, ubiquitin conjugating enzyme, and ubiquitin ligases. So activating, conjugating will do their job first, and then E3 will find out its substrate and conjugate that ubiquitin peptide to the protein that has to be degraded by proteosome. So we looked into the BRCA1 if they are ubiquitinating and degrading topo1, and we found out yes, that is the case. So we determined that BRCA1 is the E3 ligase for the topoisomerase 1 in response to topo1 inhibitor. One of my graduate students who did his whole PhD on BRCA1 E3 ligase area and demonstrated that BRCA1 is the E3 ligase for topo1 ubiquitination. These are all triple-negative breast cancer cell line. He chose to find out if BRCA1, how BRCA1 ubiquitinates topo1, and how ubiquitination is critical for the drug response. I will not go into the detail of this one. This is all published. But the other people also published that BRCA1 is critical for this pathway unknowingly before me. And that they were not able to explain when BRCA1 E3 ligase, when BRCA1 is mutated to make it a E3 ligase deficient, they realize that BRCA1 is not doing much to the DNA repair pathway. So I will take a step back and tell that BRCA1 is one of those proteins or genes that has been identified as one of the most important gene in cancer progression. They are called genome maintenance gene. They maintain the genome. So this is a very, very critical gene, and we can figure it out just by this, that around 4 to 5% of breast cancer patients have BRCA1 gene mutation, only 5%. If we include BRCA1 and BRCA2 both, we reach around 6 to 8% of total population. If we take 100 breast and ovarian cancer patients, only 8 will have BRCA1, BRCA2 mutation. So people thought it's not a big deal. But when they looked into other way around, when they looked into the patient with the person with BRCA1, BRCA2 mutation, the chances of them developing a breast or ovarian cancer is approximately 80 to 85%. That really shook the whole world of cancer research. So when they found out the couple of publications that I have mentioned here, they tried to find out that how BRCA1 is managing the genome. One of the famous publication came that BRCA1 is important in DNA repair pathway. So they looked into so one of the first enzymatic activity that was found in BRCA1 was E3 ligase function. So they mutated the BRCA1 and asked one simple question: is the mutated BRCA1 somehow responsible for DNA damage repair? They found out not. When the cells were treated with X-ray to create the DNA damage, or some other drug to create DNA damage. But when the cells were treated with topo1 inhibitor, here you can see in the right-hand panel, the cells became very, very sensitive. That means the BRCA1 is the E3 ligase, is doing the E3 ligase function for topo1 ubiquitination in response to camptothecin or topo1 inhibitor. So now we have modified our pathway where we are including that now it is phosphorylated by DNA PK 270 CoAT. Then it is ubiquitinated by BRCA1. BRCA1 does not work alone. It requires another protein, BARD1. It's a heterodimer that ubiquitinates topo1. We did a lot of work. So you can imagine a whole PhD thesis was done around BRCA1, BARD1, and in response to topo1 inhibitor. So we were really building we are putting bricks by bricks to make a small kind of strong foundation for our. So when we submitted at this time, I really wanted to publish, and when we submitted to a high impact journal, they said, what is the upstream regulator of DNA PK that is causing the phosphorylation here? By that time we had the antibody against this, and we saw that this phosphorylation is not only in response to camptothecin, but the basal level of phosphorylation is high in those cells which are resistant to the drug. So the question was, what is the upstream of this? What is regulating this one and providing resistance? So how DNA PKCS is regulated? What was the hypothesis at that time? DNA PKCS is autophosphorylated, and a lot of functions were assigned to autophosphorylation. It inactivates by autophosphorylating. Then they also reported that the protein expression of DNA PKC, where DNA PKCS is overexpressed in certain tumor types, particularly in the advanced cancers. And then third potential mechanism was upstream, there is a phosphatase, which is regulating DNA PK. The phosphatases can remove phosphate from the protein. Kinases can add phosphate group in serine, threonine, and tyrosine in general. while phosphatases can remove. So we were thinking that there is a potential that a phosphatase is involved in this pathway. And what we did is we did siRNA screening and found out some of the phosphatases. But for screening, we had a great challenge here, that how do we screen that topo1 is degrading or not degrading? So, for example, what will be my readout? We cannot go and do Western blotting and look back basically into just a static. We wanted to see the real live image of topo1 degradation. One of the challenges of topo1 is you cannot overexpress them. Cells don't like overexpressed topo1 because it fine-tunes the DNA. Cells start dying. Cells cannot survive without topo1. So we cannot overexpress. So the idea was, can we integrate a fluorescent tag just below the gene? So we dared to think about it only after the genome editing technology was almost coming into the labs. So what we do in genome editing, we use CRISPR-Cas2 set of enzyme that has been used by the bacteria for millennia. Use that and specifically target a particular gene to insert just below the gene. So genome editing has taken the world it has really changed the whole world right now. It was within 8 years of its discovery, they got the Nobel Prize for that. So here, using two set of enzyme, we can see in real time degradation of topo1. So what we did is we choose a cell line that was very, very resistant to topo1 from colorectal cancer cell line. And first found out that how many copy number of this gene is there in this cell line. And we found out 3. Gene amplification is very, very common in cancer. Particularly in colorectal cancer, it was known that topo1 high copy number is found in some of the very difficult patient population as well as cell lines. So we had to integrate this EGFP, which will fluoresce under certain stimulation of light and give a green color. So we integrated EGFP by CRISPR-Cas9 mechanism. A lab in MIT was also supposed to get the Nobel Prize for it. They didn't get it, but one of the person coming out from there was working at MGH in Harvard, and he kindly agreed to help me. And we integrated EGFP. If you can see the lower fluorescent panel, you can see that GFP is looking nice and kind of intense. But in the lower panel, when we treat with topo1 inhibitor, the topo1 intensity is gone. So here, the amount of topo1 is expressing exactly as it is in the parental cell line. So our cell line was ready. If you see, to validate that further, if you see the control here, it is nice and green fluorescent. When we treat with topo1 inhibitors, it becomes degraded in less than 3 hours. But if we add a proteasome inhibitor, it is another way to check it if it is going through proteasomal pathway. If you use a proteasome inhibitor, this intensity is not that as reduced in the middle panel. So you can quantitatively analyze, it's almost not topo1 degradation is inhibited there. So what is this telling us? That our assay is ready to screen what is the upstream event of this degradation. What is the upstream event? Can we do a screening to find out a here again, we can see in a confocal microscope here, you can see control treated with SN38, you degrade topo1 significantly. And this is a new class of proteasome inhibitor, which is in the clinic actually. Patients are treated with proteasome inhibitors for multiple myeloma. So that drug, I was working with the company that makes that drug called Takeda Pharmaceutical. We worked together to develop some new combination therapy. So anyway, here the take-home message is with proteasome inhibitor, we can basically stabilize, and this is our readout. We can read it in real time, and using this, This is another essay, basically. Using this, what we did is we looked into, there are 59 phosphatases in human genome. There are 59 phosphatases. We used siRNA library to screen what phosphatases might be upstream of the DNA PK. So when I say siRNA screen, some of you might know that silencing of a gene, we can silence a particular gene by just providing a small fragment of RNA. The discovery was very interesting. When two of the plant scientists wanted to overexpress purple color in the plant, they tried to insert a small piece of RNA or full RNA with the idea that let's see if we can insert a RNA fragment for the responsible for purple color. Can we enhance the purple color in the plant? To their surprise, the purple color, which was there already a little bit, was completely gone. So what they realized is if we provide or transduce or transfect the cell with a small fragment of RNA, or in case of plant, full length of RNA, if you can insert in the cells, it will, using a particular RNA editing mechanism, it will stop the expression of that RNA because that RNA will be chewed up by this machinery, which is supposed to edit it. So siRNA has become a very, very powerful tool in molecular biology lab. And this was also one of the major breakthrough in the last 30 years, which has given an immensely important tool to molecular biologists. So what we did is we silenced one by one all the phosphatases in the cells. In one well, one phosphatase is silenced, another well, another is silenced. And we are using this cell line where we can read the topo1 by reading the GFP expression, because GFP and topo1 are expressed together by CRISPR-Cas9. So using this tool, we are asking one simple question: what is the upstream event of DNA PK, and why it is regulated in a way that serine 10 is constantly high in this class of in the cells? Or so we did the screening, and we found out, if you see in the top is CTDSP1. We had never heard of this gene. P10, we knew P10 because P10 is very critical in brain tumor progression. So we knew about P10 for some time, but CTDSP1 was on the top. And we looked into some of these functions and we found out. Anyway, we did some work on this, but I will not go into the detail of this. But now we have modified our cartoon, or biographical abstract, sometimes they say. And then we started looking for the home for publication. So now we are saying that in the resistant cell line, the DNA PK kinase cascade is deregulated in a way that DNA PK keeps the serine 10 phosphorylated all the time. And as long as this is phosphorylated, it will degrade the topo1 very rapidly. In the left-hand side panel, if you see, the left-hand side pathway suggests that there is a deregulated kinase cascade in a resistant cell where DNA PK is constantly phosphorylating serine 10 of topo1 and degrading it rapidly. And rapid means within 90 minutes, topo1 is gone. While in the other side of the arm, everything is fine. Topo1 will not be degraded rapidly, and the topo1 inhibitor will cause the DNA double-strand break and cell death. So the right-hand panel is our sensitive patient population. Left-hand patient population are resistant to the drug. Now, as I mentioned that when we got this serine 10 phosphorylation, we right away started making antibodies and all those things. And we asked, if this serine is high, what happens in patient population? To make this possible, we knew that there are cell lines which are very resistant, and there are cell lines which are very sensitive. We raised the antibody, monoclonal antibody, and use these cell lines to see what is going on. So to make it short and more crisp, what I'm saying is in the patient population or cell line where topo 1 is not degrading, they will have lower amount of phosphorylated serine 10. That we can do by immunohistochemistry. We can check it. In the patient where these are two patient populations, these are the slides from two patients. One is a responder patient, another a non-responder patient. So we started looking at first cell lines, and then we moved into the patient population to find out if higher level of serine 10 phosphorylation is indicative of drug resistance or not. We developed this assay. We took like 2 years to develop automated chemistry, specificity, reproducibility. We did thousands of slides from the patient tumor. We used thousands of slides. We used different cancer cell line, as I mentioned, KM12, for example, colorectal cancer cell line. We used mostly HCT15, which is almost 20-fold more resistant to Colo 205. These are not my publication. It was published a long time ago. So Colo 205 in the colorectal cancer cell line is very sensitive, while HCT15 is very, very resistant. When we looked into these cells, when we made them slides, immunostain them, if you can see, HCT15 is very brown. That means it can degrade topo 1 rapidly, while Colo 205 was very blue. That means it is not able to degrade, and the cell lines are very different in their drug sensitivity. So we did a variety of things to really optimize our assay, and we call that one S10. So these are some of the slides of the patient which were quantitatively analyzed. We now started looking into a retrospective validation study of this biomarker we call P-topo1-DX. And if you see the left-hand top panel, this is the positive, and you can quantitate by digital pathology. There are scanners, you can look into that, +1, +2, +3. These are negative, and once it is quantitated, then we go to the statistician to figure it out. This is the ROC curve. My understanding of statistics is very limited, but my statistician told me that it is looking very good. In fact, the patient with higher level of phosphorylation, they are here, they will not respond. The lower level of phosphorylation, they are in this top here, left-hand panel, they will respond. So then we found out. And so we took gastric cancer patient, colorectal cancer patient slides. We annotated their therapeutic response, which is not easy to do. It's a challenge to find out whether the patient responded or not. We had to set the parameters and all. I learned a lot about who is responder and who is not, and all those things. And turned out our negative predictive value, positive predictive value, sensitivity, specificity, all are very, very good in terms of the statistical analysis and acceptability in the clinic. So we are dreaming right now how we can bring this to clinic to say some patient. Right now, it is used in the combination therapy in many tumor types where 30% patients respond and not 70% do not respond. So and there are alternative therapy. For example, colorectal cancer advanced patients receive this class of drug, and alternatively, they receive another class of drug as a first or second line. But doctors do not know till 3 months that whether the patient will respond or not. So this will be a very useful tool, not only for the oncologist, but for the patient. It is a life-saving test, basically, literally a life-saving test. So we are trying to now go to we are doing a variety of things to really bring it to the clinic, just to say the least, basically. We were able to publish this. I was very excited to publish in a clinical journal. I am showing this one because there are half a dozen oncologists who really helped me to do this work. And I hope we are particularly Kevin Hartsout. He was the oncologist in colorectal cancer area. Sinian Lee, Michael, and Al is our statistician. They were very, very helpful. They were kind of guided me through this difficult path. So we have the biomarker, and we are going into the clinic to see how we bring it to the clinic. But in next 5 minutes, I'm already little over my time. But in 5 minutes, I will tell you that how our work, which was going on for the last 2 decades, has put us in a very different situation, and which is very good for our work to move forward in a sense. So Topo1, as I mentioned, Topo1 inhibitors were approved in 2002. So in modern world, people don't want to look back, Okay, they were approved in 2002, but so what? They are old drug and the patients are getting and things like that. But so I was getting a little bit of a lukewarm response from the market, basically. Oncologists were all very excited that you have a biomarker that can help the patient, But market is very, very critical if you want to bring something to a clinic, particularly oncology clinic. So recently what has happened is there is a new class of drug that is coming into the clinic. It's called antibody drug conjugate. Here, an antibody which is expressed antibody against a particular receptor protein in the cancer. Imagine a situation where HER2, which is which drives the tumor growth in breast cancer, there is an antibody against that is being used right now to treat them. Now what they have done is they have conjugated a drug with the antibody, and that is called antibody drug conjugate. In short, we call it ADC. So in ADC, the structure is like this. An antibody will go and bind, and it will be internalized in the cell in the endosomes, and it will travel to the lysosome, or sometimes it can come out and kill the cells, basically. At that point, when it is inside, it will release the payload. They call it payload, the cytotoxic. So the antibodies are a big molecule, and while the payloads are very small, like Topo1 inhibitors are small payloads. They are 300 to 400 daltons, while these antibodies are several hundred kilodaltons, around 200, something like that. So what I'm saying is the ADCs are the emerging new therapy which targets a particular type of tumor, a particular type of cell, and delivers a drug. We call it cytotoxic payload. And it delivers, and so that it becomes specific and it becomes more potent. It took almost 30 years to develop ADCs. So if you see this left-hand side is your antibody, it requires a particular linker. And why it took so much time is linker chemistry was very critical. It's not be cleaved when we inject to the patient. In the serum, there are a variety of enzymes that are circulating that can cut it off, and the drug will be released in the circulation. So make it stable and all those things. It took a lot of time. Antibody, when the ADCs were being developed first time, antibody technology was not mature. Now they are mature. Linker chemistry is mature. And we now know that Topo1 inhibitors are one of the best payloads. So if you see this HER2 expression in normal cell, there are a few molecules out there in most of the breast epithelial cells, while in the tumor, they are expressed very heavily, and they can be targeted by this kind of antibody drug conjugate approach, and cell killing will be very high. So if you see this table, it's a very busy table, but in the top two, there are 2 Topo1 inhibitors that have been successfully being used in the clinic as we speak. And there are not many payloads which were successful. This is another class, MMA. So there are only 3 or 4 payloads which have been successfully used. Several clinical trials failed because the payload was not working very well, variety of reasons. So what I'm saying is Topo1 inhibitors are now again looked at the drug of the future, not drug of the past. And that is why we are getting some attention. Some of the people I would like to thank who worked in the lab. BRCA1 Topo1 work was done by Ankur. He did his PhD with us. He basically finished his PhD. He was an MD PhD student and finished his PhD in 3 years. Yara, Julian. Yara has gone to medicine. Julian is in management. Julian came as an undergrad, worked in the lab for several years before he went to management school. And C is also in medical school. Koji did a lot of work on particularly silencing, genome editing part. Koji came as a postdoc from Japan. He is an MD, PhD oncologist. Joe came as a medical student in the lab. And so they are all in the medicine, basically. Liz presented. So based on our work and publications, we got an invitation from some of the GRC, like Gordon Conference, and also one of my students presented in Gordon Research Conference. He did some of these 2D or 3D imaging of the topo1 BRCA1 structure. He's one of the students who came as undergrad and is in medical school this year, I think. He is a underprivileged student, came through a national screening. So we work with all level of students. That's why I wanted to tell the audience here. And this year, actually, two high school students came for a brief period of time. Acknowledgements, IHC core and cellular imaging core. Without them, we cannot work. Dana-Farber Core Institute. Dana-Farber has a couple of cores that I used. Forrest was very helpful. Jeff works on BRCA1. Susan Liss-Miller is DNA PK person. Keith Jong from MGH helped me for genome editing. Children's Hospital, Ravi is a good friend of mine. I'm working with him for some time. Al is very critical in doing my statistics for me. I would like to thank you and take any question you have. I'm really sorry that it ran over time. Any question, please?
Hello. If anyone wants to interact? Anyone wants to interact?
Shreya Singh has a question, putting up a question in chat box.
Yes, I'm seeing a question from Shreya Singh. So BRCA1, BRCA2 are tumor suppressor gene, that's for sure. BRCAD1, I understand she's trying to ask BARD1, BARD1, right? If it is BARD1, BARD1 mutations, there are two things about BARD1. First, BARD1 has been now being analyzed to understand whether they are critical for genome maintenance or tumor suppressor gene or not. But one is very well established that BARD1 is BRCAD1. I will call it BARD1, BARD1. So one thing is very sure that BRCA1 E3 ligase activity is very minimal if there is no BARD1. So once BARD1 is added, then BRCA1 E3 ligase activity is enhanced hundreds of folds, basically. So it works. BARD1 is critical for BRCA1 E3 ligase function. People are trying to understand, and there have been some report on BARD1 mutations in breast cancer patient and ovarian cancer patient. I have not looked into the recent literature on BARD1, but it is a very interesting protein in the sense that it is also it's a present with BRCA1 in E3 ligase function of BRCA1. It's a good question, though.
Hello, sir.
Yes, please.
This is Soma Roy. There is one question. Can you give us some insight into acquired and intrinsic resistance? We have studied this that lead to this reduced activity of topoisomerase in cancer chemotherapy.
Yes, it's a good question. You know, in the 90s, we were also involved in this kind of work, acquired resistance. And acquired resistance might be important in certain situations, but acquired resistance with topo-1 inhibitor, even now some people are doing work in Denmark. The Danish group that I interact with sometimes, that what they do is they gradually increase the concentration of the drug and then looked into the variety of functions of the cell when they acquire resistance. But one of the things that we have realized, and the community, scientific community in general have realized, is it does not replicate the tumor in vivo, you know. So we do culture cells in the cell lines are being grown in different concentration of drug, and they do acquire resistance, that's for sure, because of the incremental drug concentration. But unfortunately, that is not replicated into the patient population. So I will be very mindful to do a long-term project where we are looking into acquired drug resistance and put all our egg in that basket, so to say. I will be very, very careful. I'm not as skeptical. I'm not saying that that's not a good research or something, but most of the time it induces MDR, you know, and MDR overexpression perhaps overshadows some of the things. So one of the things I want to mention that it's not easy for us to understand drug resistance, particularly in chemotherapeutic drugs, because we have taken some shortcuts in the past, and we did, instead of looking into the regulation of the target drug. Unfortunately, many chemotherapy drugs do not target. We were lucky that topo-1 is the specific target of camptothecin. So looking into the regulation, we could extrapolate our findings into the patient population, which was extremely, extremely lucky, you know. So people have tried to develop predictive biomarker in our field, even including topo-1 inhibitors, and I know them. I have worked with them in the past, but they took the shortcut that we will look into gene expression profile, and we will develop a molecular signature and come up with a molecular signature, and life will be good. It didn't work, you know. So the moment it goes to the clinic, the moment it goes to the patient, things start really falling apart. Only the biology, a good biology, has to be there, basically. So I will really discourage to have a long-term plan on acquired resistance, in vitro acquired resistance, set of experiment and plan to look into the drug resistance mechanisms. I might be wrong, by the way.
Thank you so much, sir, for clearing it. Thank you.
Thank you, sir. Thank you,
Thank
sir.
you very much.
It was very informative. It was very informative in today's world scenario. Now, Dr. Hansa kindly deliver the vote of thanks. Dr. Hansa.
Hello.
Yes.
Good morning to the Honorable Speaker, Dr. Ajit Bharti, sir. And good evening to everyone, to one and all present here, respected Principal Ma'am, the respective coordinators of Department of Zoology and Biotechnology, my colleagues, and all the students present here. I, Dr. Aarti Hansda, feel honored to get the opportunity to thank you all. I extend my gratitude to the Honorable Speaker, Dr. Ajit Bharti, sir, to take out time from his busy schedule to grace the event. Thank you, sir, for your knowledgeable lecture.
Thank you. It's a real honor to chat with you all, talk to you all, and particularly the students. I always enjoy working with these students, undergrad, high school, graduate. So thank you very much for inviting me, and I appreciate this greatly. I never thought that I will be connecting with Ranchi and giving my talk from Boston. Thank you.
Thank you, sir. Thank you very much. Special thanks to Principal Ma'am for providing immense support to make the webinar special. I extend my gratitude to the organizing team, especially Dr. Salini Mehta Ma'am and Shwarnim Ma'am, to organize this webinar amidst the pandemic session. Thank you everyone for your presence. Thank you all.
Thank you.
Thank you, Dr. Bharti.
Thank you, ma'am.
Thank you.
Thank you everyone for your valuable time. Thank you.
Thank you, ma'am.